Okay, we'll get started. So last Friday we had our tour visit of the small molecule core, and today we actually have the visit to the pathology core right after class. I'll be walking people over to Building 10, and David Kleiner will be presenting his pathology slides. So today we have Neil Caporaso talking at 5 o'clock on epidemiology. Right now we have D.Salomon talking. He works up at NCI Frederick. He obtained his PhD from State University of New York at Albany. Subsequently, he did a postdoctoral fellowship at the Roche Institute of Molecular Biology in Nutley, New Jersey. And then he joined the NCI. He's currently head of the Tumor Growth Factor section. His title is Stem Cells, the Search for the Holy Grail. David.
Well, I just want to, can everybody hear me? I don't know if it's good, okay. I want to thank Terry for giving me the opportunity to give you a presentation about stem cells. My background is in mammary gland development and breast cancer, particularly how embryonic genes are re-expressed in the mammary gland, both in the embryonic mammary gland and the adult mammary gland, and how they might contribute to the etiology of breast cancer and its progression using various mouse models and cell lines from humans and also human tissues to see if we can work back and forth and get some information from the mouse. And obviously, the interest in cancer in general right now is in a large focus on the identification of cells which are involved in recapitulating the cancer and basically engendering a phenotype that is resistant to drug therapy and conventional radiotherapy and chemotherapy. And that's where the cancer stem cell theory started. It started back in the early 90s with John Dick with regard to basically identifying lymphoid stem cells and how those stem cells gave rise to the various lineages in the hematopoietic and lymphoid systems. And then in the early 2000s, Max Wisha and Irving Weissman and Michael Clark were the first to identify a subset of stem cells in breast cancer. And Hans Clevers then followed and was able to very nicely show that in colon cancer there's a population of stem cells. And in the normal colon and small intestine, they reside in a very discrete niche, the crypt. And that's one of the few systems other than the skin where we have an anatomical location to really identify these cells. That's not true for most other somatic stem cells in adult organs. So I list this in the context of saying that this is a large field with respect to regenerative medicine, not only cancer. And the fact is there are various sources of normal stem cells that have been utilized historically. And the first, and probably the most important, is the isolation of stem cells from the bone marrow in an autologous fashion to not only treat hematopoietic cancers, but also potentially stroke and multiple sclerosis and other diseases. It was then later discovered that cord blood contains a population of primitive mesenchymal stem cells, which also can have regenerative capacity with regard to the potential treatment of stroke and MS and pulmonary fibrosis. More recently, stem cells from adult organs, particularly neural stem cells, have been utilized and attempted, at least in practice in experimental systems, to treat diseases of spinal cord injury and potential degenerative brain diseases like Parkinson's disease, Alzheimer's, and ALS. And the two most recent sources, and you've probably heard a good deal about this in the news, are embryonic stem cells derived from the early embryo to regenerate entire organs, knowing in fact the lineages that actually come out of those embryonic stem cells. And then finally, and this led to a Nobel Prize by a group out of Japan, Yakanaka, they were able to identify a quartet of genes that originally exist within embryonic stem cells that you could introduce into somatic cells, not only fibroblasts, but cells of every germ layer lineage and basically regenerate a primitive stem cell called the induced pluripotential stem cell, which in many respects, but not all, resembles an embryonic stem cell that comes out of the early embryo that can be generated in that context. Cancer is a very interesting disease, and I put it in a historical perspective for you because I want you to understand my background as a developmental biologist. And Barry Pierce, in the early 70s, made a really beautiful observation. And this was actually followed up by Beatrice Mintz experimentally when she took melanoma cells and injected it into basically early blastocysts and was able to show that those blastocysts developed normally and didn't develop cancer. But what Barry showed and what her data actually formally demonstrated that cancer partially recapitulates embryology ontogenally in the sense that genes are turned on inappropriately in both in a temporal and spatial fashion. And this is not only intrinsic to the cell itself, but it's intrinsic to the environment in which the cell is exposed to. So this was the first demonstration of a microenvironmental effect way back when, okay? And more recently, in the early 2000 era, Beatrice Mintz and Mary Hendricks showed that the embryonic microenvironment in vitro, and Gil Smith, the adult microenvironment in the mammary gland, could reprogram tumor cells to become restricted to lineages which would give rise to normal cells in a normal differentiation pattern, suggesting the dominance of the niche, okay? Reciprocally, it was shown that the tumor microenvironment, or niche, could reprogram adult tissue stem cells and iPS cells to acquire properties that were similar to cancer stem cells, which are also known as tumor-initiating cells. So there's a dialogue going on here between undifferentiated cells, the transit-amplifying population that they give rise to, and the microenvironment, which we'll get back to in a minute. Now, embryonic genes are important not only in the context of normal embryogenesis and early development, but also in the context of oncogenesis, tumor progression, with respect to generating cancer stem cells or embryonic stem cells or iPS cells. And there's been a whole array of now studies trying to identify those important embryonic stem cell genes that are involved in this sort of commonality, which can function inappropriately in this two-faced fashion, in a Dr. Jekyll, Mr. Hyde sort of a circumstance, which in large part may be due to the microenvironment. Now, what about early embryonic development? And some of these genes, transcription factors, morphogens, or growth factors that are involved in regulating this. For early embryonic development, prior to implantation, starts with implantation itself, and you get the formation of the germ layers. The germ layers consist of the ectoderm, which is really a neuroectoderm because it gives rise to the neural as well as epidermal or epithelial populations, mesoderm and endoderm, and their resulting differentiated progeny. And there are a collection of morphogens and growth factors that are unique to dictating lineage specification towards these various lineages. And these growth factors and morphogens not only act in a collective fashion, but they act in a temporal fashion at specific times. It's very tightly regulated. Spatially, it's regulated within different cell types within the embryo. And there are gradients of these factors which are generated. And in fact, you can get different phenotypes depending upon the level of expression of some of these genes. And I'll give you one example. The activin nodal TGF-beta subfamily of growth factors at low levels maintains the undifferentiated state of the epiblast and embryonic stem cells, which are in fact derived from the epiblast in culture. If you raise the concentration, what happens is then they start differentiating into mesoderm cells. If you raise the concentration even further, they start differentiating into endoderm cells. So it really does show you that there's an exquisite level of control here for these factors, how they interact, and where they're expressed, and how they're and when they're expressed. Now, how do you get these germ layers being formed? Well, there has to be a separation, a spatial separation from the initial epiblast, which is the layer of the early blastocyst. And this occurs through the formation of the primitive streak. The ectoderm, which forms from the epiblast, gives rise to a population of mesenchymal-like cells through a process called epithelial mesenchymal transposition. And you'll hear more about this later. And that gives rise to mesodermal cells. And those cells then can give rise to endoderm cells through a process of MET, which is mesenchymal to epithelial transition. So it's a process that's bidirectional, and in fact is controlled by a multiplicity of pathways. And some of these pathways are reutilized in tumor cells, and this was elegantly shown by Bob Weinberg and other laboratories. For example, there is a Wnt beta-catenin notch nodal axis, which is incredibly important. So Wnt controls the expression of delta-like 1, which is a ligand for notch, and notch controls the expression of nodal. In turn, Wnt, acting through a canonical beta-catenin pathway, controls the expression of nodal, and crypto, a co-receptor for which nodal needs to function. And this is a gene that we identified several years ago and have looked at in the context of cancer and as a potential therapeutic target. Now, what about the genes that have been identified in ES cells and that might be involved in this process? Well, this is a meta-analysis from about 40 separate studies looking at common genes that are expressed in human embryonic stem cells. And I just wanted to focus your attention on 3 in particular: OCT4, which is called POOF1, NANOG, and SOX2. These 3 genes, the NOS genes, the trinity genes, are the ultimate genes that regulate early embryonic development and actually have a role in being re-expressed in cancers, particularly very aggressive cancers and in cancer stem cells. Now, these genes regulate a number of promoters and transcription factors and other genes, and they actually co-occupy the promoter regions of these genes, both in various combinations and in various complexes. So they act in exquisite concert. And there are other genes that fall into the same category, like KLF4 and MYC. Now, how do these genes function and what are their targets? Well, the NOS genes here basically activate a series of transcription factors or signaling genes that are involved in the maintenance of embryonic stem cells, shown here. Differentiated genes or tissue-specific stem cell genes are actually silenced in this context because these genes are turning on the ES cell-related stem cell genes, but the polycomb genes are basically repressor genes that are also engaged at this point and are actually repressing the expression of these genes that are genes involved in turning on tissue-specific lineage determination. And as I said, they all co-regulate each other, OCT4, SOX2, and NANOG, in a very complicated pattern, and they actually can form dimers with each other. So you can imagine the degree of complexity here and the amount of regulation that's going on in trying to understand this. So the reprogramming of somatic cells to iPS cells was shown to be due to a quartet of genes, this is the Yakanaka factors, and that original quartet was OCT4, SOX2, and KLF4 with c-MYC. But they noticed that the degree of their ability to generate iPS cells was very low, and when they did get iPS cells, a lot of the times when they put them back into mice, they got tumors. And the reason was c-MYC. So then what happened is they came along and identified other 2 genes, LIN28 and NANOG, which could replace c-MYC and getting rid of the ability of these iPS cells to form tumors and increasing their frequency to form iPS cells, which normally would have occurred at a frequency of about 0.015%, which is horrendously low. So it's now trying to find other genes which can facilitate this change, enhance the frequency, and see how they can be restricted in a lineage-specific fashion to giving rise to differentiated cell types. in vitro and then reconstituting these cells back in vivo. So stem cells, we've talked about the derivation from the early embryo, the blastocyst of embryonic stem cells and iPS cells. There are somatic stem cells that exist within various differentiated tissues. Neural stem cells, our interest in mammary stem cells, hematopoietic stem cells, and stem cells within the skin. Importantly, and with respect to this lecture, there are cancer stem cells which can either derive from normal somatic stem cells or transit amplifying cells within a particular tissue through transformation by either mutation or dysregulation of gene expression by changes within the niche, the normal niche in the tissue and the tumor cell niche, and they may not be the same. And so what you have here is a change in dialogue between cell types. So cancer is an antisocial disease. It's not just one cell type, it's multiple cells communicating at the right time, at the right place, at the right volume. So what about cancer? How does it arise from? What cell types can it arise from? Well, I've just told you it can arise from an event from a normal somatic stem cell by mutations or through transcriptional alterations. It can also arise from cells that are called progenitor or transit amplifying cells. These cells generally cycle much more rapidly than the normal stem cells, which are just generally more quiescent. Or they can arise from the mature differentiated cells themselves. So you can have cancer arising from any one of these different types of populations. And in fact, the cancer that arises from the more primitive type of cells tends to be the more aggressive type of cancer. And as you go downstream to more mature tissue cells where genetic alterations can occur, those tumors tend to be more differentiated and more treatable. So here's an example. In skin, breast, leukemias and lymphomas, colon, there are different types of cancers which arise from different compartments within each of these organ systems. In the case of leukemias, you can see here there's AML and CML, and breast cancers that derive from a primitive stem cell tend to be more aggressive. These are basal, and cancers that arise from more differentiated glandular or luminal cells tend to be more treatable and less aggressive. And the same is true in colon. You know that in the colon, Bert Vogelstein showed very elegantly that there's a transition from the normal colon to these premalignant lesions called polyps, and the polyps eventually generate tumors. And tumors can arise from any one of these populations, and the aggressiveness of those tumors is basically dictated from the origin from which these tumors arise and their treatability. So this gives you an example of that sort of lineage development and possibility of getting cancers from different types of progenitor cells. What are cancer stem cells? Do they have a specific signature or phenotype? And the question is, and the answer is absolutely, but we are still in the dark. We are still looking for signatures that on the surface of cancer cells that can be used not only to isolate cancer cells, but actually target cancer cells. So what are cancer cells? They self-renewal. They have the property of self-renewal, like a normal stem cell, and they initiate tumors at a limiting dilution, very low numbers, okay? They're regulated by morphogens that are normally involved in normal development and embryonic stem cell gene pathways. They recapitulate the original histotype of the tumor, if it's truly a stem cell. They have increased DNA repair. They have an upregulation in drug transporters. That's what makes them exquisitely resistant to conventional chemotherapy and also to radiotherapy, because most of them are quiescent and most of them are resistant to apoptosis. And one additional property which was found just recently by the Weinberg group is these stem cells, especially in epithelial cancers, can arise from epithelial cells by a process of EMT, an epithelial to mesenchymal transition. So those stem cells have properties of mesenchymal cells, not epithelial cells. And those are the bad boys, because they're the ones that are resistant and less susceptible to conventional drug therapy and radiotherapy. So what about the role of these embryonic genes in cancer and signature patterns that have been used to identify various cancers and their potential stages with regard to these subsets of genes that I've just enumerated? And here's a list of several studies showing that these stem cells, NANOG, SOX2, MYC, OCT4, some of the other ones, the polycomb gene family, show basically expression in a number of different types of cancers. And in fact, it's a wide range of cancers, and generally these genes are expressed in the more aggressive phases of the disease rather than the early phases of the disease. And that's shown here. And what we're looking at here is just a gene array display of the embryonic stem cell-like gene module that I talked about, the NOS genes, and their downstream target genes that I presented in the first table. And you can see here there's a subset of tumors that show a very high level of expression. These are very aggressive. There's some tumors that don't show that expression, but they don't show the expression of the normal stem cell gene pattern that you would find in the somatic tissue from which they arise, suggesting that there may be other genes here contributing to these types of cancers in addition to the ES-like genes. There was a very lovely study showing that in various types of solid tumors that the expression of the NOS genes, SOX2, OCT4, KLF4, CMYC, actually is found either singularly or collectively in a number of different types of cancers, and it actually correlated with tumor grade in some of these diseases. And prognosis in some cases, this is basically an early slide. It's been, obviously the information has been increased, shows that the prognosis for the expression of these genes in those cancers actually is associated with a bad prognosis with regard to patient survival and conventional chemotherapy. Now, breast cancer, that's my interest, and that's what I've been working on the last 37 years. Why this disease? Well, breast cancer is not one disease. And that's what everybody has to understand. It's really a heterogeneous disease. It consists of at least 6 different types, both histologically and clinically, of cancers. Around 250, a quarter of a million women per year develop the disease. About 40,000 die from the disease. Most of breast cancer is sporadic. About 5 to 10% is familial. And that's due to mutations within 2 genes, the BRCA1 and BRCA2 genes, which I know most of you probably have heard about. There are risk factors for breast cancer, which you should be aware of. First of all, the lifetime risk in any given woman, this is worldwide now, is 1 out of 8 women will develop breast cancer. And when it develops in a younger woman, in a premenopausal woman, it's generally more aggressive than in postmenopausal women. Why? Early menarche, which means the level of exposure to estrogen and how long you are exposed before you actually go through menopause. The other factor, nulliparity, never having children, is a risk factor because there are hormones that actually protect the breast against cancer that actually facilitate differentiation. And if you do get a cancer, potentially, it's less aggressive. That's true for people who have gone through at least 1 pregnancy, but multiple pregnancies, there may be some increase in risk, but that's still an open-ended question. And another very interesting observation, and I think this is absolutely fascinating, and it relates to the microenvironment, that obesity, that is the adipocyte content of the breast, especially in premenopausal women, increases the risk of breast cancer by 70%. Adipocytes are not just making fat guys. They are making adipokines, cytokines and growth factors that actually contribute to the breast and its development. So the more that you have as far as adipocyte content, the greater your risk is, potentially in a premenopausal woman where she's also exposed to estrogen. So what about breast cancer? Can we see this same stratification with regard to the expression of these NOS genes and their targets in a clinical setting? Well, if you look at grade 1, 2, and 3, this is going from a very low-grade type of tumor to a high-grade tumor. You see that the high-grade tumors generally express the NOS gene targets relative to the grade 1 and grade 2 tumors. You look here, the red meaning high expression, green meaning low. The same is true of estrogen receptor status. ER-positive tumors tend to be more treatable, more differentiated than ER-negative tumors. Again, these tumors tend to associate with the expression of these NOS genes and their downstream targets. And the same is true for the molecular subtype. Remember, I told you there's 6 molecular subtypes. There's luminal A and luminal B. These are very differentiated tumors. They have ER and PR expression, estrogen receptor and progesterone receptor expression. There's a normal subtype, which is somewhere in between that, and it's either expressing one or the other receptor, so it's not both receptors. There's a HER2 positive subtype, which expresses ERB2, and then there's a basal population, which consists of at least 3 different types. There's a basal, a metaplastic, and a Claudin low population. These are bad news, guys. These are untreatable breast tumors that metastasize and don't respond to any form of conventional chemotherapy. These are the bad boys. These have the highest level of expression of these NOS genes and their targets. And if you actually look here with regard to survival, overall survival, and metastasis-free survival, there's a direct correlation with respect to the expression of these activated ES signatures in both of these settings. So it does have clinical significance, trying to find out how these genes function. Not to beat a dead horse, but here's stratification based on stage of cancer in the breast. And I've listed NANOG, OCT3, 4, and SOX2 here, and this was taken from a large study. And you can see as you go from the early stage cancer to the late stage cancer, the level of expression of these genes rises dramatically with regard to the stage and the degrees of aggressiveness. Now, what is the breast? How do you study it? It's very difficult to do this in a human setting. You can get mature breast, you can get breast tissue from reductive mammoplasty tissue, but is that really normal? Probably not. So people have turned to the rodent as a model system, which we have done as well. And there are various stages of breast development in the rodent that occur after birth. There's embryonic development, but that's another story. But most of the development of the mammary gland in the rodent, the mouse and the rat, is postnatal. In the virgin gland, basically you have a very simple architecture of arborizing ducts consisting of primary ducts, secondary ducts, and tertiary ducts. And up to about 3 to 4 weeks, there's not really much going on. But after 3 to 4 weeks, estrogens and progesterones kick in. And then what you start to see is hyperbranching. And if the animal goes through pregnancy, okay, with progesterone levels and prolactin levels rising, you get the development from these terminal structures of alveoli during pregnancy and lactation, which basically produce milk proteins. Now, in the breast, like other tissues, it's not just the epithelial cells, which is very low in the virgin. It rises about 1,000-fold, which is massive, going from the virgin to the lactating breast. But there's adipose tissue, there's stroma, there's endothelial cells, and there's infiltrating macrophages, leukocytes, and neutrophils, which also contribute to the development of the duct-like structure in the breast, okay? And in the duct itself, you have the luminal cells, which line the interior of the duct, and the myoepithelial cells, which are almost like smooth muscle cells, which basically surround the outside of the duct. And if we look at this in more detail, this is a GFP-labeled mouse. This is in the virgin. You can see the ducts. These are the terminal end bud-like structures that I was pointing out that eventually become alveolar structures during pregnancy. And within these structures, there's subpopulations of cells. There's a cap cell population that sits around here that basically contains the mammary stem cells, the most primitive stem cell population in the mammary gland, which can give rise to all of the other lineages within the gland. Within the terminal end bud, there's a population of more epithelial cells that are less mesenchymal. These are called luminal progenitor cells, and they give rise to the mature luminal cells here, whereas the cap cells give rise to the myoepithelial cells here. So there is a method to the madness, and there's a sort of a substructuring of architecture and histology that you have in the breast, but it's not as clear as in the colon or in the skin where you have a bulge-like population or a crypt cell population. Now, there are various players between these populations which control the normal breast development. Okay, so let's look at the luminal progenitor population sitting within the terminal end buds. They respond to progesterone and estrogen. There are effector proteins that are turned on by these systemic hormones that are involved in not only controlling the luminal progenitor population proliferation and lineage, but also control the basal population that I was talking about, the cap cell population. As an example, Wnt4 is a growth factor that activates the Wnt receptor, and it's induced by progesterone and activates a Wnt receptor in the basal population. So it's a paracrine effector. It's one population affecting another population in a paracrine manner. And Wnt4 can induce delta-like 1, which is a ligand for Notch4, which sits in the luminal population. So here you have this feedback and control system where two populations are talking to each other through two different effector systems. Okay, and that's just giving you an example as to how this whole system is intricately balanced. The same is true for the rank system. The rank ligand is expressed basically in the luminal population, but its receptors expressed in the basal population. It itself, again, is controlled by progesterone. So here you have a superseding systemic control of local growth factors that are controlling the dynamics of how cells are talking to each other. What happens in cancer? In cancer, this paracrine dialogue system breaks down. Okay? What happens is these cells don't talk to each other anymore. They start making a lot of these growth factors, and they start feeding back on themselves. So it's an autocrine system. And then all hell breaks loose. And so now you've got to understand what the circumstances are here with respect to the breakdown in this communication system and who's important in the communication system and at what particular stage of the cancer. And that's not an easy task. How do you assay for a stem cell in the context of a mammary gland? Well, you know in the bone marrow, you can do bone marrow transplantation and bone marrow replacement, and you can measure bone reconstitution of the bone marrow. The same is true in the mammary gland. You can actually clear the mammary fat pad of the intrinsic population of epithelial cells, let it sit for a week or so, and then what you do is you introduce a heterologous epithelial population from another type of mammary cell or species or genetically altered mouse into that cleared fat pad. And what happens is you will get outgrowth from that population that you've reintroduced. So you've reconstituted the mammary gland from that recipient donor population into a host. And you can genetically tag both the host and the donor, and you can ask who's important in developing not only the normal mammary gland, but tumors and premalignant lesions. So what are some of the markers that have been used to identify tumor cancers in the human? CD133, which is otherwise known as prominent. CD44, that's the hyaluronan receptor, if you're not familiar with it. There are two drug transporters which have been utilized as markers. And there's aldehyde dehydrogenase, which has been recently shown to mark stem cell populations in various cancers. This is an interesting gene. This takes aldehydes, okay, and converts them to acid. And what is it doing? Actually, it's taking retinol and converting it to retinoic acid. And retinoic acid is a differentiation factor. So mutations in this gene or alterations in its expression may have a significant impact with respect to maybe the retinoid system and how it's used in a clinical setting. What about the mouse? Well, in the mouse and the human mammary gland, there are a unique set of markers that have been identified that can be used by FACS analysis, fluorescent activated cell sorting, to isolate different subpopulations of stem cells or progenitor cells. A number of these markers, interestingly enough, turn out to be integrins. CD29 is beta-1 integrin. CD49F is alpha-6 integrin. And CD61 is beta-3 integrin. And again, CD44 is the hyaluronan receptor. So you can see that there are different markers here, both in the mouse and in the human, some of which are common, some of which are unique to these species, but they do subserve a purpose in isolating different, both primitive stem cells and progenitor or transit amplifier populations. So how do we do this? How can we measure, oncogenetic potential of cancer stem cells, and particularly in the breast? Well, you can take either a primary tumor, a breast cancer cell line from either a mouse or a human, or a mouse xenograft, okay, from either a transgenic mouse or an artificially xenograft setup. And you can use FACS sorting using these various markers to isolate different subpopulations depending upon how you gate, and that's an important criteria, and ask which of those populations have the ability to reconstitute the original tumor from which these cells were derived, or the cell line, at a high frequency but at a low dilution. And so you can either do in vitro assays, you can look at sphere formation, called tumor sphere formation, and this was derived from the original idea about using neurospheres in the nervous system to look at stem cells. And you can do serial passaging of these spheres, looking at their ability to self-renewal over time. And the longer they are capable of self-renewing as you passage the spheres, the greater the probability is that you have a high percentage of stem cells, okay? The in vivo correlate to this is to take those cells that have been FACS sorted and you do a limiting dilution assay. You take the cells, you put it back into an orthotopic site, in this case the mammary gland, and you ask the question, what's the fewest number of cells that you can use to get the tumor? And is the tumor that you get the same type of tumor from which the cells were originally isolated from, histologically and by the use of other markers, phenotypically? And obviously, serially passing those cells to see if that property of self-renewal is basically reproducible. Now, in the mouse mammary gland, as I've told you, there's a primitive mouse mammary stem cell, which gives rise, these are quiescent, to a transit amplifying or common progenitor cell. These are more rapidly dividing. These cells give rise basically to a myoepithelial progenitor or a luminal progenitor population. The luminal progenitor population can give rise to either alveolar cells during pregnancy, or they can contribute to the normal ductal epithelial cells seen in the non-pregnant gland or in the pregnant gland that don't differentiate and secrete milk proteins. The myoepithelial progenitor cells give rise to, obviously, the myoepithelial cells. So you have a lineage here, like in the hematopoietic system, okay? And there are various markers that have been identified to segregate these populations, okay? And there are various mouse transgenic model systems that have actually been developed. Overexpression of Wnt1, p53 null mice, BRCA1 null mice, activation of Notch1 or 4, ERB2, polyoma middle T, RAS, and MYC, which actually arise from different subpopulations along these lineages. So each one of these models offers its own specific advantage to maybe correlating this to the human disease as far as different types of cancers in a clinical setting, and trying to understand the molecular phenotypes associated with these types of mouse tumors and how you might be able to treat those tumors and understand those tumors. By the way, if you don't know, basal tumors, they're also called triple negative. I don't know if anybody's ever heard of this in the literature. Triple negative tumors are basically ER negative, PR negative, and HER2 negative. They're nasty. And that's what's shown here. Basically, again, in the human, you have this lineage that I've just described for the mouse. It's very similar. And you have cancers that can arise from these different lineages. Claudin lobe tumors are incredibly bad news. They're resistant. They arise from the primitive stem cell. The biprotein progenitor cell rising from BRCA1 mutations, which arise basically in a luminal progenitor population, give rise to a basal-like tumor. The HER2 and luminal A and luminal B tumors generally arise from luminal cells at later stages in differentiation. And as you can see here, these more aggressive tumors are more mesenchymal, and these guys here are more epithelial or luminal, the more differentiated ones. So cancer origin. Okay, cancer cell. You got multiple lineages from which the cancer stem cell can arise. Mechanistically, how does this occur? And this observation by Weinberg was really very, very seminal. He showed that either the primitive stem cells or the luminal progenitor cells could give rise to cancer stem cells, okay? And what he showed was that if you were to induce epithelial mesenchymal transition, and there are a variety of factors, which I'll get to in a minute, that can do this, the cells that arise basically have acquired a stem-like feature. They're more mesenchymal, and they exhibit properties of stem cells. They also acquire very unique properties which make them highly metastatic and aggressive, okay? Again, they're mesenchymal, they're resistant to drugs and anchoesis. They're extremely migratory and invasive. They basically are resistant to senescence and apoptosis, and they're resistant to immunosuppression. So that's one unique path or biological pathway, which, by the way, is a developmental pathway that's engaged in cancer that's giving rise to potentially to cancer stem cells. So here we have it. We have epithelial cells, mesenchymal cells. You have EMT driving it this way. You have MET driving it that way. There are a number of structural proteins that can be used to characterize the epithelial phenotype, some of which I've listed here. These are tight junctional proteins, adherence proteins, claudins and occludins, and some extracellular matrix proteins. Likewise, the mesenchymal cells also have a series of structural proteins like vimentin and encoherin and fibronectin and smooth muscle actin, which are used as markers, as well as a number of transcription factors that are engaged in the mesenchymal cells that are not engaged in the epithelial cells, like snail and slug and twist and goosefoot and FOXC2. These are all factors that are basically EBOX binding transcription factors that repress a number of these epithelial-like genes, and then also engage the expression of these mesenchymal genes. And what are the factors regulating this? Well, there's a whole slew of factors. Hypoxia through HIF-1 alpha. You have a number of different types of growth factors, signaling pathways, and changes in mirrors that can trigger these pathways, either EMT or MET. And it's trying to understand which are the important players and how can you use this in a therapeutic setting to interrupt this process. And I should say, in the primary tumor, when it metastasizes, it has to go to a mesenchymal cell. But then when it gets to the metastatic site, it converts back to an epithelial cell. So it has to go through an MET. Oh. So what about in the mammary gland? You have normal breast, you have hyperplasias, you have carcinoma in situ, you have invasive carcinoma and metastasis. So you have, like I just said, EMT, when the cells basically intravasate into the circulatory or lymphatic system, and then they extravasate into either the lung or the bone or the brain, and they undergo MET. And there are a number of genes in the breast that have been identified which are important in each of these processes. And I'm just giving you an overview here without trying to dwell on this. So one of the genes, and this relates to our work that we've been excited about, is crypto, which is a co-receptor for the nodal GDF family of TGF-beta-related proteins. It's a GPI-linked glycoprotein that can function either in cis, attached to the cell by a GPI anchor, or in trans as a soluble factor. And it's cleavable, and it's an adapter and chaperone protein, not only for nodal and GDF1 and some of the TGF-beta family members, but it's also acting as a chaperone for Wnt signaling and Notch signaling. So it really is a multifunctional protein and has different functions in different contexts. And without dwelling on it, it was first identified as a gene that it was expressed in early embryogenesis that was necessary for inducing anterior-posterior axis formation, primitive streak formation, and laterality. It's expressed in the postnatal mouse mammary gland in a subpopulation of cells. It's expressed at low levels in most somatic cells. It's expressed at a very high level in a number of different human carcinomas. And it induces EMT and migration and invasion and a whole number of properties associated with very aggressive cancers. And like I said, we have now ventured our way into using this as a potential therapeutic target with a biotech company developing an antibody that's been conjugated to a microtubule disrupting agent to target this cell-associated protein. So what does it do? And in that list, here's just some experimental data. If you take a mouse mammary epithelial cells, they're very cobblestone in appearance. If you overexpress crypto or treat them with recombinant protein, they become very mesenchymal and they start scattering. You take these cells and you put them into a type 1 collagen gel, unlike the normal cells that form these very rudimentary-like ducts, these things form really very beautiful arborizing ducts that are actually hollow and have a lumen. And actually, we've developed several transgenic models using crypto, using either using conditional mammary gland promoters like the MMTV promoter or the WAP promoter. And what happens is you get hyperbranching, you get the development of premalignant hyperplasias, and eventually these develop tumors. So it is oncogenic and transforming both in vivo and in vitro and in vivo. And what did I do here? I turned it off. What is it? Is it associated with any of the different molecular subtypes that I've talked about in breast cancer? And if you can just focus on the purple bars here, here's luminal A, luminal B, HER2 positive, and basal-like. And you can see that the expression of crypto in these tumors goes up dramatically, especially in the HER2 positive and basal-like subpopulations of breast cancer. So there is an association there with this particular gene, this embryonic gene. And what about other types of tumors? And this is a Herculean study, and this is not just my lab. This is about 20 different labs that have looked at multiple types of tumors and the frequency of expression of this gene, either by immunohistochemistry, real-time RT-PCR, or by microarray analysis. And you can see that in a number of these tumors, actually in a majority of them, this surface antigen or surface molecule is expressed and suggests that it might be, in fact, a viable therapeutic target. So, in concluding, we focused on the cancer stem cell. What about the niche? The niche is equally important. We're dealing with a dynamic system here. This is, a dialogue going on here. And the dialogue is not only between the tumor cell, whether it's a cancer stem cell or a transient amplifying cell or a fully differentiated tumor cell or a cell that's metastasized, but it's dealing with cells in those sites that are talking to those tumor cells. In the case of the primary tumor, there's something called CAFs. These are carcinoma-activated fibroblasts. These are different from normal stromal fibroblasts that sit in the normal breast or other tissues. These are fibroblasts that exhibit desmoplatic-like changes and fibrotic-like changes. And they're different from their normal counterparts. There are tumor-associated macrophages that are different from circulating macrophages. Okay? There's endothelial cells that are dumping cytokines like VEGF and other things like angiopoietin. And then, to make it even worse, there are bone marrow cells coming into the environment, mesenchymal stem cells, and myeloid progenitor cells, which are contributing to this nightmare. And I'll get to the players that these cells basically secrete. These cells undergo EMT. They have to then reverse their phenotype after they get into the circulation and undergo MET to recolonize back in a secondary site. So you have a tumor niche, you have a metastatic niche. There are cells in this niche, and I'll talk about that in a minute. There's also a circulatory and lymphatic niche. When the cells get out into the hematopoietic system or the lymphatic system, there are cells there talking to those tumor cells and supplying cytokines. So in the primary tumor and the metastasis, you see all of these different players, and the bone marrow is contributing, the local environment's contributing. Here are some of the players that are basically contributing to exacerbating either the primary tumor or maintaining the phenotype of the metastasis that basically is derived from the primary tumor. And actually, interestingly enough, the primary tumor sets up the secondary organs before the tumor cells actually get there. So they prime that secondary organ system, the seed and soil hypothesis, to prime it. They set up a fertilizer system so that those cells now have a preference of colonizing specific sites. We don't know why that happens and why certain of these cytokines or ancillary cells home to specific end organs, secondary organs. And here comes the real hook. Circulatory system and cell types, not only from the endothelial cells, but circulating cells that dump cytokines, extracellular matrix proteins that basically feed the tumor cells in the vasculature and the lymphatics. And now out in the metastatic site, I don't care where it is, the brain, the bone, the lung, you've got effector cells here, myeloid cells, macrophages, fibroblasts, mesenchymal stem cells. These are secreting all sorts of lymphokines, cytokines, and growth factors that are exacerbating the growth of the metastasis and leading to secondary metastasis, okay? And here are some of the factors listed here in a more detailed fashion, and I'm not going to go into it, but you can see in some of the pathways that are involved here, it's really quite extensive. And so I've been trying to give you an overview of this. Can we use this in a therapeutic sense? How can we use cancer stem cells as a potential target? Well, most of conventional chemotherapy and radiotherapy has been treating the primary tumor, the fully differentiated tumor. That's been an absolute disaster in most cases, except for some oddball cases like, embryonal carcinomas, because these are already differentiated. And it's the stem cells which are quiescent and resistant to these drugs and radiotherapy that really should be the target, okay? So we have to find appropriate ways of targeting the cancer stem cell population and the progenitor cell population, as well as how do you prevent metastatic spread of these bad boys to secondary sites. And what's in the secondary sites that's facilitating the tropism of those cells towards those secondary sites. So here are some of the novel pathways that are being looked at by big pharma and biotech companies. Wnt pathway signaling inhibitors here, either against the ligands or the receptors or the transcription factors. Notch pathway inhibitors. There are gamma secretase inhibitors, which are involved in processing of the Notch receptor that can hedgehog pathway inhibitors, such as cyclopamine, and this is one of Genentech's big targets. There are basically inhibitors against the TGF-beta activin nodal pathways, such as inhibitors of the type 1 receptor ALK1 or ALK tyrosine serine threonine kinases, or ligand or receptor blocking antibodies that are being developed to uniquely target these cancer stem cell populations. There also now is an interest in targeting the cancer niche. So it's a double whammy. Maybe not only should we should be hitting the cancer stem cell, but the surrounding supporting cells within those either primary tumors or metastatic sites. And in a normal setting, you have a supportive cell. You also have non-cellular elements like the extracellular matrix, which is contributing to the maintenance of these tumor cells. And then in the cancer niche, you have an entirely different set of parameters, possibly, that are being regulated. And here's a list that has currently been looked at as potential therapeutic targets, at least in the bone marrow, but this is also true in other metastatic sites for treating the niche and targeting the niche. So I hope in this talk I've tried to give you an overview as to the complexity of the system and the fact that, conventional chemotherapy and radiotherapy is basically, right now, I think, witchcraft. We need to know mechanisms. We need to know molecular pathways. We need to know how these cells basically dialogue with each other and what are the molecular pathways of communication between these various cell types. Thank you. Do all cancers have stem cells? Yes. Different types, but yes. That's a pretty well-accepted theory at this point. Everybody once thought that, it was only lymphoid hematopoietic malignancy. Not true. Without a doubt, all carcinomas and sarcomas. And that's interesting. And that's why the sarcomas are really worse than the carcinomas, because they're already mesenchymal. So their stem cells are bad news. Chondrosarcoma, osteosarcoma, nasty. Yeah, good question. Good question. Actually, people are looking at BMPs, bone morphogenic proteins, as a way of taking basically mesenchymal cells and pushing them backwards towards epithelial cells, okay? Actually, it's interesting you ask, because BMPs in the lung are necessary for maintenance of the lung metastasis colonies. So in that context, you need a BMP inhibitor. You know what I'm saying? Because those are the guys in the lungs that are basically maintaining the epithelial phenotype, but it's the opposite in the primary tumor. So if you look at COCO, an inhibitor of BMP, if that's expressed, it's going to suppress metastasis. If you knock it out, and there was a beautiful Cell paper on this, the primary metastases explode and they give rise to secondary and tertiary metastases. So it's all context dependent with respect to whether it's the MET or the primary tumor. But yeah, they're looking at both sides of the, yes. What percentage of the cells? It varies from cancer to cancer. You can have from anywhere from 0.01% up to 1 to 5%, depending upon the type of cancer. Okay? And that's all, that's in flux. I mean, that's all based upon the markers that we have today that we're using to isolate those cells. And it's also based on the animal models that we're using to identify cancer stem cells. Example, using a nod-skid or a nude mouse. You know, the frequency of cancer stem cells was 0.01 to maybe 0.05 or 1%. And then Sean, I forget his last name, but up in Michigan, used a nod-skid IL-1 gamma mouse. And the proportion of cancer stem cells that they could detect in that background went up to 10 to 15%, because it's more immunocompromised. So it's system-specific, I mean, as far as the host that you're using to assay, as well as the markers that you're using to identify. Thank you, David. You're welcome. Wired. Arthritis is a bitch.
Okay, our next speaker is Neil Caporaso. He got his MD degree from the University of New Jersey. Subsequently, came to NCI as an oncology fellow in the medicine branch. And now he's chief of the pharmacogenetic section, division of DCEG. And he's going to talk to us today about epidemiology.
Okay. Okay, I have been given my orders, and this lecture is going to be done before 6. Well before, so I'll give you all a chance to grill me if you want. Epidemiology. Okay, we're part of NIH, you know that, part of the National Cancer Institute, and my division and my branch are part of the intramural program. As you know, 85% of the money's extramural, so we're a minority. And in NCI, our division is Cancer Epidemiology and Genetics. We just got a new division director, Dr. Stephen Chanik, took over for Dr. Joseph Framini, who'd been there for 30 plus years. And our mission is to understand the cause of cancer. And in our group, I'm a branch chief of the genetic epidemiology branch, but we have other branches that focus on nutrition, the role of hormones, the role of infections and immunity, role of occupation. We have a group that solely focuses on statistical methods, and we have a group that is interested in radiation and cancer. So my outline, introductory concepts. Epidemiology refers to the causes of cancer in the population. Different from clinical medicine that focuses on individuals. There's not an epidemiology of cells. Really, this is groups of humans. And a distinction is that it's an observational science, not experimental. We don't get to do experiments on people. We don't get to pick who's exposed and unexposed, something that happens in life. People make choices about what they're exposed to, directly or indirectly. We have to, therefore, worry a lot about bias, and a big issue in epidemiology is controlling bias. The goals of epidemiology, we want to identify the causes of cancer. That's our group in particular. We want to quantify risks and risk groups. We want to understand something about mechanisms, and this is a trend in epidemiology. We want to calculate the need for public health and health services and identify syndromes. And just to contrast, again, observational versus experimental, we're ethically prohibited from doing experiments on people. So we observe populations and we look at their outcomes and we relate them to what people do. More and more we relate them to genes, genetics. And this weakness of epidemiologic studies, this observational component, was used by the tobacco companies to try and say, you know, it's not really science. It's, you know, we don't believe this stuff about tobacco and lung cancer. And so I'll go into that a little bit just to make that point. Epidemiologists emphasize prevention. And the rationale for this is that it's very, very effective. And if you really want to save lives, of course, you don't want to worry about therapy. You really are much more efficient if you prevent disease. So it's effective, it's cheap, it has a public health orientation, and it eliminates disease at the source. The problem, though, is that it's difficult to demonstrate the effectiveness of epidemiologic interventions. It's less dramatic than treatment. And when you go to Congress and you've got a cure, it's really much more dramatic and more politically effective than to say, you know, if I clean up the water 20 years from now, you know, the rates of all these diseases are going to go down. So because you don't have people on TV that appear and say, I was cured by this intervention, what you just have are people without disease. So the lives saved appear in statistics, not grateful patients, and it's sometimes harder to get funding, which is derives from the political impact. Also, it's hard to get Nobel Prizes. So in spite of the immense work it took to assemble the data to show that tobacco caused lung cancer, none of those individuals got Nobel Prizes, although you could argue that among all of them, they probably saved the most lives. So again, that's just the way it goes. Anyway, there's primary, secondary, and tertiary prevention, and I won't go into that. Epidemiologists, you can't hang around epidemiologists too long without hearing them talk about bias. And I'll just say that this is systematic deviation from the truth, and there are all sorts of bias, but a particular one is that when you do an epidemiologic study, if the participation rate is low or it's not representative, you have a problem because the group that you're studying is not representative of your target population and therefore not generalizable. And so selection bias is a key kind of bias, and I'll talk about that a little more as we go forward. So we did a study in Italy, and we asked for funding for a large epidemiology study where we were going to collect tissue and blood so we could look at genetic factors and biochemical risk factors. And site visitors came, and of course, if you're in the extramural world, you have to apply for grants and you get the money. If you're in the intramural world, you get a certain amount of money, but every 4 years the site visitors come and review what you're doing. So the site visitors said, we are not going to allow you to do this study unless your participation rate is very, very high, over 60, even 70%. So we did a phone survey and said, how many people in this population will participate in the study? It's a 1-hour questionnaire. We take 70 cc's of blood, and you're really doing a lot of good for humanity. And 30% said they'd participate. So then we gave them a letter. We followed up by phone. We told them we would study them in the hospital or at home. We gave them a cash award. We got a letter from their physician saying it would be a great thing if they would participate. And that went up to 49%, still not good enough. We would not have been able to do the study. So then we got new interviewers. So these were nurse interviewers who were very charming, and the men really liked them. And we had male interviewers for the women, and that was great. We got the physicians to call them up. Instead of money, we gave them a gas coupon. This was Milan, Italy, and gas was really hard to get. We had TV ads at night. We got a new invitation letter. The mayor, who was actually a popular mayor, we got a letter, and we got a toll-free phone line. And so that got us up to a sufficiently high participation rate that we were able to do the study. So we did that study back almost 10 years ago, and we've had over 40 publications from that study. It was a great success. So when epidemiologists do studies, they worry a lot about the controls, and population controls, like I just showed you, are expensive, but they're the most representative. And the big worry is selection bias, that you can have perfect population controls, but if you're only getting 1 or 2% of your target population, there's a big worry that that 1 or 2% are very non-representative of the group that you want. And most studies, unfortunately, have convenience controls, and this can really bias the results. So population controls are the gold standard. They have to be representative of the population from which the cases derive. You can calculate absolute disease rates, and you reduce selection bias. Okay, so if you ever decide to do a study, an epidemiologic study, and you call in an epidemiologist, the questions they're going to ask you are, what's your study design? Where did your controls come from? Did you collect key covariate data? Covariates refer to the factors that can influence disease, and you need to capture data on them. So for example, if you're studying lung cancer, you really have to also collect data on family history, on smoking, on diet, and on occupation. Bias and confounding, what was your original hypothesis as opposed to data dredging? What was your power? And how did you validate your markers? And a common question epidemiologists get is, can you tell me why my grandmother, who smoked all her life, never did any exercise, had a terrible diet, was an alcoholic, outlived all her doctors? And again, the answer to this is that it's a probabilistic science, not deterministic, and you always have outliers. Okay, some tools that epidemiologists use, and I will go through this pretty quickly. Cancer maps. So we've created maps, by we, I mean our division, of different conditions. And this is very helpful because one glance at this map can tell you something about the reasons for different kinds of cancer. So a tier of higher rates of melanoma across the southern United States tells you something about melanoma, mainly that sunlight is a risk factor. This has progressed. There's geographic information systems that now, with satellite information, can get exquisite information on patterns of diseases and exposure. SEER stands for Surveillance, Epidemiology, and End Results. And it's a gathering of data across about a quarter of the US population. And it collects data on incidence, survival, demographics, and various data about tumor sites. And it's a comprehensive source of population-based information. So you can go online and get all sorts of great SEER data. And you can see tables and figures like this. And here's I'll just show a few points about SEER data. Here are cancer rates for men and women. And you can see there's a peak here in men and no corresponding peak in women. Does anybody know what that comes from? Yes. Exactly. So this was the introduction of PSA screening for prostate cancer, which caused an artifactual rise in cancer prostate rates as more people were screened and more disease was identified. It's unclear if the disease that was identified actually was helpful. It remains a controversy to this day, but you can see the other rates for cancers remain stable. Here are the rates for women. And you can see that if you just looked at all cancers, there was a bigger rise in African-American men than white men because the rate of prostate cancer is generally higher in blacks than whites. Here you can see a diagram from 1975 to 2000, and the death rates, the mortality rates, appear to be leveling off a little bit, or at least plateauing in men and women. Does anyone know why that is happening? Any guesses? Okay, I'll show you. That is because of lung cancer. The most common cause of cancer death is lung cancer, and it's leveling off. And is the reason more effective treatment? No. The reason is the decline in tobacco use. So here's cancer death rates in lung. And since about 1984, when they peaked, they're going down. And in women, they peaked later, but now they are going down also. And just to show you, in children aged 0 to 14, incidence rates are much higher than mortality rates. And this generally means that we have a cancer, a rare cancer, where treatment is rather effective. Okay, well, given the problem I mentioned with observational science, how do epidemiologists prove a cause? Or how do you assemble evidence in favor of a cause? And a classic paper back in 1965 established that there are a number of things that you should consider to gather evidence for a cause. One is that it's helpful if the factor that you believe is the cause confers a high risk. Secondly, it's helpful if, when you conduct different studies with different designs, the risk factor is consistently seen. A third very important factor is a dose response. If you have more of the risk factor, you'd like to see more of the disease. Fourth factor, temporal order. You want to see that the cause occurs before the disease. And fifth is biological plausibility. You want the biology to make sense. So here's data from 3 cohorts. And this is an amazing amount of data, actually. And it shows lung cancer rates in relation to cigarettes smoked per day. And these 3 cohort studies, each of them well over 100,000 individuals followed over time, the lung cancer rates are remarkably consistent. So that's a beautifully consistent demonstration that tobacco is associated with lung cancer. And then a second factor is the dose response. It's pretty linear. So that, again, is a highly supportive sign that tobacco is the cause. Here's the temporal aspect I showed you. So you have male smoking, which rose in the 1900s through the 20s, et cetera. And this was followed by a rise in male lung cancer. And socially, female smoking began to be more acceptable in the years prior to World War II. And then in the 60s, it really took off with advertising campaigns that targeted women. And sure enough, if rates of female lung cancer followed. And here's the biological plausibility. And this was a study by Oscar Auerbach, who at Tulane and then later at New Jersey did studies of beagles where he attached them to smoking machines. And he was able to show that the progression of cellular changes from metaplasia to dysplasia to carcinoma in situ occurred in a way that paralleled the histologic changes that were observed in humans. So that was a nice demonstration of the biological plausibility of the tobacco association. And finally, these are the relative risks of lung cancer when you do smoking cessation. So it takes about 20 years to approach the baseline when individuals quit smoking. Now, the risk for cardiovascular disease goes down much faster. It goes down in a year or two. But lung cancer takes a number of years, but it does steadily decline. So that's another nice biological plausibility. One of the unknown mysteries of lung cancer is what precisely is the dosimeter of damage. In other words, why does the damage persist here? The morphologic changes associated with lung cancer regress after about a year or two, but something is still causing elevated risk years down the road, and that molecular dosimeter would be a very interesting thing to identify, but we don't know what it is. Okay, accomplishments of epidemiology. Epidemiology is responsible for the identification of many specific causes of cancer, and epidemiologists advocate for public health and prevention. And again, I'm sticking to the example of tobacco because it's probably the best documented. And increasingly, epidemiologists want to have a role in the molecular redefinition of cancer. So the general risk factors for cancer are pretty well known: increasing age, environmental factors, increasingly genetic factors, and combinations. So this crude diagram illustrates what used to be thought of as the causes of cancer. Tobacco is a good third, diet perhaps another third, and then a mix of other factors. If you look at the data, however, all of the data, you can make the argument that virtually all cancer is ultimately due to the environment. So I work in a genetic group, and so we often have to talk about this, but the fact is that really it's mostly the environment. So I'll just touch on the evidence for this. One piece of evidence is international variation in cancer rates. And if you go down the list of tumors and you look at the countries where the highest rates contrast with the lowest, the ratio of the highest rate incidence rates to the lowest is always fairly dramatic. So some of this could be due to genetic factors, and that argument is often raised. But if you do migration studies where you have people in the right age group that migrate from one country to another, typically after a short period of time, they acquire the rates of the country that they've migrated to. And this supports an environmental component. So here's an example. Jingwei is a province in China, and you remember the cancer maps I showed you before. And this particular province has the highest rates of lung cancer in China. And why is that? Well, if you go there, what you find is this. tremendous rates of indoor and outdoor pollution. So they have these beds, these ovens that heat up the beds and produce smoke, called kangs. They cook indoors, and the volatile oils, the indoor pollution, and the outdoor pollution combine to make this the highest rates of lung cancer in the world. This is a copper smelter in Montana, and I don't know if I put it in, but there's a cancer map, which you could imagine. And if you look at this cancer map of lung cancer, in Montana, there's a red line, actually a red area in Montana where this copper smelter was. And the copper smelter basically was contaminated with arsenic. And so the area around the smelter was a very high rate of lung cancer. And when the smelter was removed, the rates returned to normal. So tobacco is the big cause, and tobacco is still the #1 cause of morbidity and mortality in the United States and worldwide. 1 in 5 US deaths, 10 million deaths per year, 30% of all cancer due to tobacco. All of the tobacco-related sites are difficult to treat. The cost of tobacco-related disease is staggering, and this persists in spite of widespread knowledge of the health consequences of smoking. If you give people questionnaires in the United States, virtually more than 90% of adults are aware that smoking is bad for them. When we did our study in Italy, we gave a questionnaire to the individuals who participated because we wanted to know, are they aware of the dangers of tobacco or not aware? Because it requires a different public health intervention if you're not aware. But in Italy, they were also aware of the dangers of tobacco, and yet they chose to smoke, or more likely, they were dependent on tobacco, and so they kept smoking and were unable to quit. So here's the per capita consumption of different forms of tobacco in the United States, and this shows the impact of epidemiology. This the peak rates of smoking through the 50s and 60s. And here you had the Surgeon General report, which consolidated the evidence and established in a forceful way that tobacco was the risk factor. And this took a long time to have an impact, but slowly over time, it began to have an impact. And through the 70s, you started to get the secondary benefits of social strictures on smoking. And this led to studies of environmental tobacco smoke. The first studies showed that never smoking women, spouses of smokers, were at higher risk, a study by TRACO in the early 1980s, and then increasing studies that showed that environmental tobacco smoke was a risk factor. Finally, a Surgeon General report in the 1980s. And these studies were critiqued, but essentially meta-analyses confirmed again and again that both workplace exposure to tobacco smoke and exposure in the home was a risk factor. And so this, in turn, led to clean air legislation, the elimination of cigarettes sections on planes, elimination of smoking in hospitals and schools. And that, in turn, helped further reduce the rates of smoking. So one of the things that this made us realize is that for a lot of risk factors, for instance, it's emerging more and more that sugar, and particularly soda, is a risk factor for a lot of diseases. So it's going to take a lot of scientific study and then a lot of social changes to conquer that risk factor. It'll probably take decades. But even though it's a difficult thing, that's a pathway to eliminating a lot of diseases that are related: obesity, diabetes, et cetera, and possibly cancer. So the second risk factor after tobacco is alcohol, and it's related to a number of cancers. Ionizing radiation is an important risk factor. And I show you one slide on this, but the amount of work that's been done in these areas is staggering. many studies of ionizing radiation and cancer in relationship to the A-bomb explosions in World War II, medical exposure, radionuclide exposure, occupational exposure to radiation, environmental exposure. We have a group that follows the Chernobyl exposure and shows, higher rates of thyroid cancer, and which appear to be persistent over time. And one of the major discoveries of this group is that for the first time, there's a dose response for chronic lymphocytic leukemia. And chronic lymphocytic leukemia is the most common adult leukemia in the United States. And after the atomic bomb, that data there was examined, and there was no increase in chronic lymphocytic leukemia. So if you study, the dogma was always that acute myeloid leukemia was related to radiation, but not CLL. And in fact, that may have been wrong, and it may have been that CLL is rare in Asians, and so there were insufficient numbers in the atomic bomb cohort, but in Russia, there are sufficient numbers of CLL, so they were able to detect this increase. I mentioned that non-ionizing radiation, UV radiation, is important for skin cancer and melanoma, and tanning beds are clearly a risk factor. We have a group that studies immunity and infections and cancer, and there are a number of infections that are clearly related to different kinds of cancer, and newer hypotheses about different viruses. Some of these are well established. For example, HCV clearly is related to elevated rates of hepatocellular cancer and NHL. H. pylori and stomach cancer. One of the major discoveries and accomplishments of epidemiology is establishing that 100% of cervical cancer is related to HPV. And more recently, cohort studies have established staggering risks associated with HPV for oral pharyngeal cancer. And this is new work establishing a relative risk and odds ratio of over 200 for oral pharyngeal cancer. So it's clear that HPV has a role in a number of other cancers, also anal cancer. We did a study of HPV and lung cancer and found no association. Just mention that. And newer technologies are being used to identify heretofore unsuspected infectious agents. So this is just an example of sequencing conducted in colorectal cancer that two different studies that identified Fusobacterium nucleatum, and there are some newer studies that also implicate this organism. Newer and microbiome studies are investigating novel infectious agents. So we may actually discover some unsuspected infectious sources of cancer. Not insignificant are occupational carcinogens, and actually the majority of extrinsic environmental causes of cancer are environmental, are occupational. It's long lists of them. And an example, these studies are very difficult to perform because they're politically fraught. So a diesel study that was done by the Occupational Studies Branch in collaboration with NIOSH, found a significant role for diesel exhaust and lung cancer. And this study was reviewed by Congress a number of times, and well-funded industry groups fought this study, and now they're spending $1 million and using a FOIA, a Freedom of Information Act inquiry. They have asked for all the data of the study, and they will try and rip it apart. But very likely, they will not succeed. It's pretty clear that diesel exhaust is a risk factor. Similarly, formaldehyde. So these studies are incredibly important to do. They're very hard to do extramurally. You need a stable group that can actually assemble the expertise and conduct these kinds of studies, because if we don't do them, I'm not sure who's going to protect the public by identifying these as risk factors, so the appropriate safety measures can be taken when these kinds of chemicals are used. Okay, challenges of epidemiology. Well, I told you a little bit about the political challenges. There are a lot of other challenges. One is that there are a number of cancers for which we don't know the environmental cause. We believe it's out there, but we don't know it. How exactly diet contributes to cancer has proved incredibly elusive to figure out. So is it major food groups like carbohydrates, fats, protein? Is it vitamins? Is it bad stuff in food? You can make the case for each one of these hypotheses. For example, aflatoxin contaminating peanuts is clearly a cause for a small segment of liver cancer, but it's likely not much of a cause for colon cancer or the major cancers that are thought to be related to diet in the American diet. Similarly, I haven't said much about genes and gene environment, but basically we understand the genetic cause for a number of rare familial cancers, and we understand from genome-wide association studies that there are common genetic factors that contribute to common cancers, but we're only able to identify a few percent of what we believe is the total heritability. So there are some genetic mysteries lurking that we can't explain. Population studies are expensive. They take a long time to conduct. Sometimes the results are controversial, and there's a lot of junk science out there to refute. So if you ask the public, there are still many people who believe that autism is caused by vaccination, and believe other clearly false or ideas that have been pretty much refuted by high-quality studies. But that's a problem. So in the categories of cancer causation, there's the environment and genes. And gene-environment interaction is an oft-postulated idea, but there are very few examples of documented gene-environment interaction. I'll show you one in a minute. So I already said on the environmental side, there are some cancers for which risk factors are unknown. When we know the cause, what causes individual susceptibility is poorly understood. Gene environment is poorly understood. Some causes are poorly studied. So what do I mean by this? I told you we've studied, done a pretty good job at studying infectious diseases, some occupational carcinogens, alcohol, tobacco. What hasn't been studied? Well, we studied diet a lot, but it's very difficult to precisely quantitate diet or study it in a molecular way. Everyone agrees that food diaries are incredibly difficult to calculate, and food frequency questionnaires are fraught with inaccuracy. So that's a tough one. Another tough one are areas that are difficult to capture. Physical activity, physical inactivity, sleep, circadian variation are highly plausible causes for cancer that are almost not studied because it's very hard to get at them. You want to study circadian variation? Okay, we can study night shift workers, or we can study airline pilots. But essentially, you're assuming that because they have that occupational label, their circadian rhythm is disrupted, which is, as we say, a problem because there's a lot of misclassification bias in making that assumption. So we really don't know. If you transplant tumors into mice where you disrupt their circadian variation by ablating their pineal gland or the suprachiasmatic nucleus or blinding them or doing all these things that disrupt they're a circadian pacemaker, they get a lot more tumors, or the implantable tumors grow faster. So there's a lot of evidence, but it's something that has been inaccessible to descriptive epidemiology. So chronic lymphocytic leukemia is an example. No environmental cause known. I told you that radiation may be a risk factor. Another possible risk factor are herbicides, but essentially all of this leukemia is quite common. We don't understand the environmental cause, and we don't understand what makes it progress. So I told you about the difficulties with dietary risk factors. We looked at diet in lung cancer. There's a lot of problems here about what might be the dietary component to lung cancer. The idea of nutrient-based interventions for lung cancer was a spectacular failure in that the alpha tocopherol beta-carotene trial in Finland, you may have heard of it, 30,000 Finnish men roughly were randomized to get beta-carotene or alpha tocopherol. And the alpha tocopherol vitamin E did nothing. The beta-carotene increased the rate of lung cancer. So, wow, millions of dollars down the drain to actually cause harm. So, then there's the idea that our processed food is bad. There's something about the processing and we need to return to more traditional food, but we can't return to that traditional food because our ground is depleted of nutrients. There's the problem of obesity. There's the question of how to aggregate nutrients. And then there's meat and vegetable consumption. And we focused on meat and vegetables. And in fact, we showed that both fresh red meat and processed meat increase your risk of lung cancer after taking into account every other risk factor for lung cancer that we know. We also showed that vegetable intake was somewhat protective. Okay, genetics. Let me say, and I'll just cover a few more items, and then I'll stop and let you ask me any questions. New technologies in the past half-decade have really accelerated genetic discovery, as many of you are aware. And by this, I mean GWAS technology that came into common use around 2007, 2008, where you would do 200,000, then 500,000, then 1 million, and now 2.5 million and 5 million common SNPs, and then in a large group of cases and controls, try and associate those SNPs with disease. So that has identified some risk factors for common cancers, and we have identified a few familial cancer genes, but overall, we only identify a small portion of the variation and very little gene environment. So there's a lot to go on the genetic side. Often, people counter the idea of the environmental cause of cancer by saying, no, it's genetic cause because every single tumor is accompanied by genetic changes. And as you're aware, likely, TCGA, the Tumor Cancer Genome Project, with the papers that are coming out, has identified tens to hundreds of genetic mutations in tumors associated with cancer. One of the problems is that we had hoped that there would be a handful of specific mutations associated, and in fact, it's not a handful. There's kind of a large group, and there's a lot of work on trying to sort out the so-called driver mutations from the passenger mutations, and that's a challenging task statistically, and a lot of folks are working with sophisticated computer programs to try and derive the tree diagram that will show the original mutation and then the different clones. And so in some way we can understand what the precise origin of the tumor is. Nevertheless, it's hard to infer the specific clinical behavior of tumors from the 100 or so mutations that you see. And of course, for different tumors, melanoma and lung cancer have the most mutations, and then there's some of the leukemias that have the fewest. Nevertheless, it's a little bit of a challenge to interpret the existing data, so it's going to be very exciting to see how that data evolves. So of course, when we talk about genetics of tumors, there's both germline and somatic mutations, and there's a big contrast between family and population data. In families, and we study high-risk families, where cancers follow a Mendelian pattern, we often can find one major gene that accounts for a specific tumor, whereas in populations, there's not one gene. There are many genes that additively increase or decrease your risk. The argument that all cancer has a genetic component derives from family studies and studies of large populations where you look at the family history in cases and controls. And when you do that, for example, in lung cancer, and there are more than 20 such studies, you invariably find that having a family history increases your risk of the tumor after accounting for every other risk factor that you can quantitate. Everything from gender to residence, where you live, education, all the details of your smoking habits, whether your spouse smokes, et cetera. So that's the area of genetic epidemiology. And here are the kinds of high-risk families that we study. So we have a family where we actually have 3 cases of CLL and a case of non-Hodgkin's lymphoma. So this is where we study rare genes. And we have over 200 such families, and we work in a consortium. And we've done a variety of different kinds of genetic analyses. And I have to tell you that we still have not isolated the gene that accounts for CLL. But we're working on it. And here's examples of cloned genes for different familial syndromes, just to give you some examples. And here's what you find in GWAS studies, those genome-wide association studies that are conducted in large populations. So pretty much all over the genome, we've identified different genes. In some cases, the same gene for multiple tumors. So there's a few examples. And in some cases, it's very hard to sort out what's going on. So in lung cancer, it was very exciting because one of the first things that was discovered when the first study of this type was done was that a gene that we had never studied as a candidate called the nicotinic receptor on chromosome 15 was identified. And so that was exciting because it's highly plausible, a gene that alters the ability of nicotine to bind either in the lung or in the brain sounds like a great gene and a plausible one that would contribute to lung cancer. The problem is that it appears that the mechanism is that it increases nicotine dependency, makes you more likely to smoke. So may not be acting on lung cancer directly, may be acting on your brain to make you inhale the cigarette deeper, hold the smoke longer, and therefore absorb a larger dose of carcinogens, not directly related to the tumor process per se. Okay, so I think I will stop there. Here, I'll give you one example of gene environment in lung cancer, and that is N-acetyltransferase. And this is a gene that detoxifies aromatic amines. Aromatic amines are compounds that are in dyes. And in the late 1800s, it was noted that German dye workers working with those bright aniline dyes would get a lot more bladder cancer. And so it's one of the first occupational carcinogens known. So aromatic amines are bladder cancer carcinogens, and they cause bladder cancer, but they're also metabolized in the body by this N-acetyltransferase 2. And in the general population, about 50% of us are slow acetylators, we're slow at detoxifying it, and the other 50% are fast acetylators. If you are a slow acetylator, the aromatic amines hang around longer and have more of a chance to act as carcinogens. So in smokers, if you have this gene, you're at much higher risk than if you don't have the gene. And this gene actually was detected in a GWAS study. So it was a candidate gene first, and then it was agnostically discovered among 200,000 other genes. So this is a pretty solid example of gene environment, but there aren't many others. Future for epidemiology. So I told you that in spite of the fact that lung cancer is going down, still we get more cases of lung cancer in former smokers. So we told people to stop smoking. They stop smoking. They still get lung cancer. Treatment is still a problem, even with the molecularly targeted mutations. So we understand that EGFR related tumors can be treated with a class of medications, and ALK-related tumors with another class of medications. In spite of that, that's only a small percentage of all lung cancers. And essentially, therapeutically, the survival rate for lung cancer has only improved, the 5-year survival rate, from 13% to 16%. Maybe it's a little closer to 20%. So we have a long way to go. And I will touch on 4 avenues of going forward in the next 2 minutes to conclude. Traditional epidemiology like to look at exposures and related to disease, but molecular epidemiology collects biospecimens and looks at stages on the pathway to disease. So internal dose, early biological effect, altered structure or function, and then disease. And so the study I told you about in Italy, we actually went into the operating room, we would get tissue, and we would have the pathologist dissect it, and we would put it in liquid nitrogen. And so that tissue is very, very valuable today. So we had 2,000 lung cancer cases, and about, considering that about a third of them were operable, we have about 500 fresh tissues and about 900 tumor blocks. And I told you already we looked at meat very carefully, so we had a good epidemiology questionnaire in Italian. And molecular epi studies of this kind have had a great success rate. They were able to show that 100% of cervical cancer is due to HPV. We were able to show clearly that cutting down smoking is ineffective, because when you cut down, your body doesn't really care. It wants to extract the nicotine, and so you absorb more carcinogen. You need to quit smoking. And GWAS studies that have identified genes for 100 or so conditions would never have been done without the collection of blood, biospecimens, and tissue. And finally, integrative epidemiology is the latest stage, and that's where we have a link to translational medicine, because we actually don't just focus on disease. We actually collect information on outcome, whether individuals live or die and how they respond to treatment. This is the whole area of pharmacogenomics. And we also collect behavior, and I could go into what we've gotten out of that. But I think I'll stop here and ask if anyone has questions or areas that you want to know more about. Yes?
You mentioned how people, I don't think they were at risk, but you are there strategies to avoid the to get formed or to actually, the part of the problem?
Yeah. So you're probably aware that when people try to quit smoking on their own, there's the point prevalence of success at 1 year is only about 5% or even less. So a number of strategies have been used to encourage smoking cessation. Many of them focus on nicotine replacement therapy and the use of antidepressant class medications like bupropion. The latest and most innovative approach uses varenicline, which blocks the nicotinic receptor. So that receptor I told you about, that variation in that receptor is actually related to smoking. If you block that and you use nicotine replacement and you use counseling and you use an antidepressant, you can approach 45% success rate at smoking cessation. So there's a tiered group of interventions, and as you use each one, you improve the smoking cessation rate. But more and more people feel that a multimodal approach, layered approach, with social interventions as the bottom line. So you can't smoke at work. You educate people at school. You have your spouse yell at you if you try and smoke in the house. You get offered free counseling at work. So there's layers and layers of social layers. You know, you put ugly pictures on the cigarette packs. You raise the tax. An economic strategy is very effective in certain groups. So it doesn't help you if you are upper middle class and you're 50 years old. But if you're a teenager, the economic cost actually is somewhat helpful. Unfortunately, a big impetus on smoking among teenagers is your peer group. So the fact that your parents don't smoke doesn't help. And the early social measures, you know, where you'd have that sign in the store that says if you are under 18, you are forbidden to buy cigarettes. Well, for teenagers, that was like, I got to buy cigarettes, you know. You're not gonna, you have to have a strategy targeted, like you show that they have ugly teeth or that you're sexually unattractive. You know, teenagers need something totally different. And if and it's their peer group or the group just a little bit older than them. So for kids in high school, they look at college kids, and that's what they imitate. And if they see a movie where a college kid is smoking, you know, that's more effective or in a bad way than anything else. So that's, I don't know if that answers your question. Yes?
In regards, going back to the secondary risk factors for complex syndromes, I mean, how do you deal with issues that, like with the red meat thing? You know, one might argue that people who red meat have other additional behaviors that expose them to increased risk factors. That has nothing to do with eating red meat, but it just happens that that group segment. How do you deal with those?
So that's a really good question, and that's a big problem in epi studies, and that's one of the reasons that the meat association is somewhat controversial. So it's been long known that people that eat red meat also tend to smoke. And so the simple response is fine. We have detailed smoking information on our questionnaire, and we're going to adjust for smoking in the analysis and take that into account. The problem is that you have to have a very fine-tuned questionnaire because you're going to get residual confounding. For instance, if I just divide people into non-smokers, former smokers, and current smokers and adjust and say, I'm done, I've taken smoking into account. In fact, you haven't, because among your current smokers, the meat eaters have smoked for a longer duration, they smoke their cigarettes further down, they take more puffs, they smoke more cigarettes per day, and they're more addicted. If you ask them the key question, when do you smoke your first cigarette? Within 5 minutes, 5 to 30, 30 to 60, greater than 60, the meat eaters tend to smoke more in the first 5 minutes. So even if all the other smoking parameters are the same, they're more addicted, they get more carcinogens, and that may account for the meat effect. So one thing we do is, by looking at non-smokers, if your questionnaire is good and they're a non-smoker, you pretty much got a non-smoker. and you can eliminate that form of bias. And so the meat data tends to hold up even in non-smokers and to a good degree in former smokers, and after as careful an adjustment as we can do. But it is still an issue, and there may be other correlates like education level with ill-defined correlates like urbanization or exposure to air pollution or blue-collar occupations that don't necessarily have defined lung cancer carcinogen exposures, but in general, you're under bad conditions and it's dirty and maybe, you're just getting multiple minor exposures that cause it. So I agree it's controversial and it's difficult to deal with.
I mean, epidemiology by itself, all the results and outcomes are they all tell statistics behind it. And these results can be clearly associated with it. Like one can go very aggressive on that, say, I'm going to be with a p-value of pull down interview, or maybe you can go to a p-value of 0.25 or 0.1. But one is smoking is a very example because very high p-values can be achieved by not at other things. Is there any particular convention? Because one at the percent of 0.45 one can make a statement, and then 0.01 one can make a statement, 0.01 is more powerful. Is there any terminology observed in the video about community to take more powerful statements?
So that's a great point, and there's a vast debate about this and a vast literature on this, and a lot of the early genetics work, for example, was criticized because people would find a 0.05 p-value, which is the typical convention, and but they would test 50 genes, and they wouldn't take into account multiple comparisons. So I think people are much more aware of that, and particularly in the genetics area, a very conservative correction, a Bonferroni type correction, where you take the number of hypotheses and you divide it by the p-value. So typically for these genome-wide studies, the p-value has to be 10 to the minus 8, 5 times 10 to the minus 7 or 10 to the minus 8, depending on how many genes, to achieve a level of statistical significance. But then it has to be taken somewhere else and reproduced. So almost everybody says, we almost don't care what the p-value is. Take it to another independent study and reproduce it for us, or we don't believe it. So that's really the current, you can almost pick your initial alpha level, whatever you want. We still want to see an independent verification in another population. And I think that I showed you those criteria and the consistency that no matter what the initial p-value is, reproduce it independently. And typically for the second study, people are willing to accept a p-value at a more modest level. So if you come to me and say, I don't know, telephone wires are a risk factor, fine. You have an initial study. Now, take it to another country or another study or a different study design and meet that 0.05 value, one-sided, fine, and it'll be convincing.
We'll have to close now. Again.
Thank you.